Hello, my name is Joshua Gabi. I welcome you to Photosynthesis. Our lesson objectives, today we are going to look at, we shall look at photosynthesis, we shall look at light spectrum, we shall look at chlorophyll, chromatography, action and absorption spectra, photosynthetic stages, and limiting factors. You are welcome. Please do not forget to subscribe. Photosynthesis. Photosynthesis is the process by which certain cells synthesize carbon compounds, for example, glucose, from inorganic molecules, that is carbon dioxide and water, in the presence of sunlight energy. So we are saying that photosynthesis is a process by which certain cells, they synthesize carbon compounds, and the major one is glucose, and it is synthesized from inorganic molecules, that is carbon dioxide and water, in the presence of sunlight energy. That process is what we call photosynthesis. We use carbon dioxide. It is combined with water in the presence of chlorophyll, which traps sunlight energy, and we get glucose, and then we also produce oxygen as a byproduct and also some water. So that whole process is what we refer to as photosynthesis. This process requires photosynthetic pigments, and the photosynthetic pigments we always talk about is chlorophyll. So the whole process of photosynthesis, it requires photosynthetic pigments, and that pigment is chlorophyll. Plant cells have specialized organelles adapted for this function. So the specialized organelles that we find in plant cells that are adapted for this function of photosynthesis, they are called chloroplasts. The chloroplasts are found in chlorophyll. They're the ones that have got chlorophyll, and chlorophyll is the one that traps sunlight energy. So we want to look now at photosynthesis versus cell respiration. Photosynthetic organisms, they use sunlight energy to create stored chemical energy. So we have the photosynthetic organisms. These ones are going to use sunlight energy, and when they use sunlight energy, then we shall form what we call stored chemical energy. We have our sunlight energy, as the source of energy, and this one will be trapped by chlorophyll, which we can find in the chloroplasts, and inside here now we shall convert the sunlight energy into chemical energy, that is in the chloroplast. Now the energy is stored in carbohydrates. That energy is stored in carbohydrates and is released by cell respiration. We have seen that the energy that is coming from the sun, that energy is converted into chemical energy in the chloroplast. Now, after being converted into energy in the chloroplast, now it is this energy which is stored in the carbon compounds is the one that is going to be released by cell respiration. Now, in cell respiration, what is going to happen is that the carbon compound will be taken in as glucose. Now, this one will react with oxygen, and that reaction is going to take place in the mitochondria. And when that reaction is taking place in the mitochondria, we shall end up producing products like carbon dioxide and water. They will be produced. But the major substance that we are going to produce is going to be the energy, which we are now going to use. Remember, that energy we are saying, we got that energy from the sun. We got it from the sun, and then it was stored in carbon compounds in the chloroplast. Now from there, of course, the organisms will take in the carbon compounds, and then they will take in the carbon compound. The major one is glucose. This glucose, in the presence of oxygen, will be the one which is broken down in the mitochondria. In the mitochondria, then we shall release our energy, which is going to be used by the cell. But also, we shall also release waste products like carbon dioxide and water, which will now be taken back and used by photosynthesis. Animals cannot synthesize this compound and must consume them as food. So as animals, they cannot, of course, synthesize the compounds, which are the organic compounds. So they just consume them. So what synthesizes those organic compounds is the chloroplast. The chloroplast will absorb, of course, that light energy, and it will use this light energy to build up the organic compound, which we refer to as glucose. And it is this glucose which cannot be synthesized by animals. So the animals will consume it, and when they consume it, now they will use it in the presence of oxygen in the mitochondria, and then they will break it down to release that energy which has been stored in the carbon compound by the chloroplast from the sunlight, and then it will be used as energy in the cell. And then the waste products that are produced, carbon dioxide and water, will be taken back to the plant to be stored again as energy in the form of glucose. Let's look at the light spectrum. Now we have the visible light, and that visible light which comes from the sun is composed of a range of different wavelengths of different colors. For example, if you look at our spectrum here, we have the red, the orange, yellow, green, blue, indigo, and the violet. That is our visible light, which is coming from the sunlight, but with different wavelengths. It runs from 400 nanometers to 700 nanometers. Now the spectrum of the visible light ranges from 400 nanometers, that is the violet light, to 700 nanometers, which is the red wavelength. The colors of the visible spectrum are from the longest. The one which has got the longest is the red with 7 nanometers, to the shortest wavelength, that is violet with 400 nanometer. That is our visible spectrum. It runs from the red, which has the longest wavelength of 700 nanometer, to violet, which has the shortest wavelength, that is 400 nanometers. The visible light has a range of wavelength which is from violet, which has got the shortest, and the wavelength of red, which has got the longest, all of us that we have seen. If you look at your spectrum, you'll see that violet has the shortest wavelength, which is 400 nanometer, and then the red light is the one which has got the highest wavelength, which is 700 nanometer. That is the visible light. Let's look at chlorophyll. Photosynthetic pigments are used to absorb light and convert it into chemical energy, that is ATP, which is an intermediate, or glucose where it is stored. So we have the photosynthetic pigment, and remember the photosynthetic pigment that we are talking about is chlorophyll. Chlorophyll is the photosynthetic pigment which is used to absorb sunlight energy and convert it into chemical energy, which we refer to as ATP, which is the immediate source of energy in the cell, or glucose, which is stored in the plant. The main photosynthetic pigment in the plants is chlorophyll. That is the main photosynthetic pigment. The main photosynthetic pigment in the plant is chlorophyll. For example, if you look at our plant here, you can see the different plant cells inside, and you will see those green color. Those green color, that is those are chloroplasts. And those chloroplasts, if you look at them, they are green in color because they have got the photosynthetic pigment, which is chlorophyll. Chlorophyll absorbs the red and the blue. Chlorophyll absorbs the red and the blue light most efficiently. It absorbs the red and blue light, which is most effectively absorbed effectively. That is chlorophyll. And the green light is reflected. That's why we are able to see the green color, because the green light is reflected. Therefore, the green color of the leaves are green because green light is not absorbed by the plant, but it is reflected. It absorbs, chlorophyll only absorbs the red and the blue light most effectively. Other accessory pigments, like for example carotenoid, may absorb other wavelengths of light, and this is to maximize the rate of photosynthesis. So we have other pigments which are not chlorophyll. We have seen that chlorophyll, for it absorbs the red and the blue light. But we have other pigments like the carotenoids. These ones, they absorb the other wavelength of light. And this one is mainly to maximize the rate of photosynthesis. Chlorophyll, for it absorbs the red and the blue light most effectively, and it reflects the green light more than other colors. So chlorophyll will only absorb red and the blue light, and it will reflect the green light. That's why we always see most of the plants are green in color, because that green color of light spectrum is reflected, and we are not able to see the other colors because those ones are absorbed. There are other pigments like carotenes. These ones now, they absorb other colors which are not absorbed by the chlorophyll, so that they can maximize the rate of photosynthesis. Let's look at chromatography. Chromatography is a technique used to separate pigments. Chromatography is a technique used to separate pigments. The fluid is passed through a static material, and that static material is what we call a stationary phase. You pass the fluid through the static material, which is the stationary phase. And now the pigments that are dissolved in a fluid are going to move through. They will move through the stationary phase. So we have our chromatography as a technique which is used to separate pigments. For example, if we look here at our chromatography here technique, we have a fluid. Down, we have a fluid here. There is a fluid here down, and this fluid has pigments dissolved in it. And then we have the stationary phase or stationary material, which we have here, this stationary material. Now it is dipped, it is placed in the fluid, the tip of it is placed in the fluid here, and then the pigment will start rise, the fluid will start rising in the stationary phase. As they are traveling into the stationary phase, then the pigments are going to be separated. You will see different pigments being separated at different positions or different distances. Now the pigments, these ones are going to move at a different speed. They move at a different speed, hence they will be separated. Those are our pigments. So you can see as the pigments are moving from the fluid phase, they will enter into the stationary phase, and as the fluid is moving up in the stationary stage, because the pigments are moving with different speed, they will start to separate out at different points. The retardation factor can be calculated of each pigment. Each pigment, we can calculate its retardation factor. For example, we have different pigments. We have a green pigment here. We have a blue pigment here. We can calculate its retardation factor. We have a yellow pigment here. All these ones we can calculate their retardation factor. The orange pigment is here. We can calculate their retardation factor. And when we're calculating the retardation factor, we can get the distance the pigment has traveled. We get the distance the pigment has traveled. For example, if you look at this green pigment, it has moved from here up to this point. So we can get that distance traveled by that pigment over the distance the solvent travels. Now, if you look at the solvent, it has traveled up to this point. So we shall get the whole distance of the solvent from up to down. And then we get the distance of the pigment traveled by the pigment. So for the green, we shall get this pigment and we place it on top. And then we get the other pigment of the solvent, we put it down. So we shall get the distance traveled by the pigments over the distance traveled by the solvent. Now that one will give us our retardation factor. So the retardation factor is calculated for each pigment, whereby we get the distance that is traveled by the pigment and you divide it with the distance traveled by the solvent. Then you get your retardation factor. Let's look at the action versus absorption spectra. Now this is our action spectrum. This one shows the light wavelength used by pigments, especially for photosynthetic rate. Here we are seeing that the rate of photosynthesis is highest in the blue. If you look here, it is highest in the blue action spectrum, and also towards the red side. That's where we have the highest action spectrum in the wavelength of the blue, which is, and then also the wavelength of the red. Now the absorption spectrum, this shows the wavelength absorbed by the photosynthetic pigments. The absorption spectrum shows the wavelength absorbed by the photosynthetic pigments. Now we have the photosynthetic pigment, there is chlorophyll B. We have chlorophyll B. This pigment absorbs the green light. It absorbs the green light mainly in much of the lower waves. And then chlorophyll A, it absorbs the yellow light. It is absorbing the yellow light. Then we have the carotenoids. These ones are absorbing the orange light. Let's now look at the stages of photosynthesis. Stages of photosynthesis. Photosynthesis occurs in two distinctive stages. It occurs in two distinctive stages. Now the stages we are talking about, we have the light-dependent stage and then also light-independent stage. Those are the two stages in which photosynthesis occurs. Light-dependent stage or light-dependent reactions convert light energy into chemical energy. That is, the light-dependent reactions, they convert light energy into chemical reaction. So here where we have the light-dependent, we are going to get water, which is going to go inside, and also light. So the light, it is going to take place in the thylakoids. It will take place in the thylakoid, in the chloroplast. This is a light-dependent reaction, and it absorbs sunlight energy and converts it into chemical energy, which is our ATP. Then the light-independent reaction or light-independent stage. This one uses the energy to synthesize carbon compound. So we have our light-independent stage or light-independent reaction. This one is going to use the energy which was made from the light-dependent reaction or stage. It will use this energy and combine it with carbon dioxide, then we shall produce the organic compound. So those two stages of photosynthesis, the light dependent uses sunlight energy to produce energy, ATP. And then the light independent stage or reaction, it is going to use that energy, which is from ATP, from the light-dependent reaction. We'll use it to combine the carbon dioxide to produce organic compounds. Please don't forget to subscribe. Let's look at the light-dependent reactions. Light-dependent reactions. Now the light-dependent reactions, these ones, they occur in the thylakoid membrane of the chloroplasts. Those are the light-dependent reactions. They take place on the thylakoid or in the thylakoid membrane, which are found in the chloroplast. So when sunlight energy is absorbed, this sunlight energy can be used to split up water. It is going to be used to split up water into hydrogen and oxygen. Oxygen now is going to be released as a waste product. Now the hydrogen will be taken by the hydrogen carrier, and then it will be also, we shall produce ATP energy. So light is absorbed by chlorophyll and resulting in the production of ATP. This light is going to be absorbed by chlorophyll, as we have seen. And now when it is absorbed by chlorophyll, it is going to be converted into chemical energy. This chemical energy is the one that we are storing as ATP. Then the light, which is also absorbed by the water, which is split to produce oxygen and hydrogen. Now that same light, still which is absorbed, is going to be absorbed by water. And water will now produce the hydrogen, and it will also produce the oxygen after splitting up. The ATP and hydrogen are used in the light-independent reaction. So we are seeing that the light energy is used in the light-dependent reaction, and it is used to split up water to give us the hydrogen. Whereas at the same time, that light is absorbed by chlorophyll, and it will be converted into chemical energy, which is ATP. Now, this ATP and hydrogen are the ones now that are going to be used in the light-independent reaction. So that's why we are saying ATP and hydrogen on ADP-H are used in the light-independent reactions. Let's now look at the light-independent reactions. Light-independent reactions occur within the stroma. They occur within the stroma of the chloroplast. We have looked at the light-dependent reactions. We said they occur in the thylakoids of the chloroplast. But now the light-independent reactions, these ones, they occur within the stroma of the chloroplasts. Stroma is like a cytoplasm of inside the chloroplast, the fluid-filled space in the chloroplast. So that's what we call the stroma, and that's where the light-independent reactions take place. The ATP and the hydrogen are transferred to the stroma of the chloroplast. Remember, in the light-dependent reaction, we produced the ATP and we produced the hydrogen, which is carried by the NADP. And these ones, when they are produced in the thylakoid, they will be now transferred to the stroma in the chloroplast. Now, in the stroma, when they diffuse into the chloroplast, now they will now be used to combine with carbon dioxide, and then they will be used to fix carbon dioxide, and we shall end up producing our organic compounds. Organic compounds will be produced. But these organic compounds are produced by the use of the hydrogen, which was produced in the light-dependent reaction, using the energy which was also produced in the light-dependent reaction, combining it with carbon dioxide, and then we shall fix carbon, and then we shall form our organic compound. And that one is taking place in the light-independent reaction. The hydrogen is combined with high carbon dioxide, and this one will form a complex organic molecule. So we have our hydrogen, which is going to combine with carbon dioxide, and when they combine together, they will form a complex organic molecule, which is our end product. And that one is taking place in the stroma, and it's what we call the light-independent reaction, because it does not depend on light. So the ATP will provide energy that is required to power the anabolic reaction. This ATP here is the one that is going to provide the energy required for the whole reaction to take place. It is an anabolic reaction because we are going to end up building up the organic compound. Building this organic compound is going to come from carbon dioxide, using also the hydrogen that we produced in the light-dependent reaction, and then we shall fix the carbon, and then we form the organic compound. So the ATP which has been provided will provide energy which is required to power this anabolic reaction. Let's now look at the limiting factors. Limiting factors. We are seeing that we are producing organic compounds. Organic compounds are produced by using sunlight energy, which is converted into ATP. And after being converted into ATP, which is taken to the light-independent reaction, where it is used up with the hydrogen, which we got from splitting water, and it is used to fix carbon dioxide, and then we form organic compounds. Now we want to see the limiting factor. The law of limiting factor states that when reaction depends on more than one conditions, the rate of reaction is limited by the factor that is nearest to its minimum value. So if we are having a reaction which is depending on more than one conditions, the rate of that reaction will depend on the factor that is nearest to its minimum value. So if there are very many factors affecting a reaction, the factor which is mainly going to affect that reaction is going to be the factor which is nearest to its minimum value. Now, photosynthesis is dependent on a number of favorable conditions, and those conditions are the ones that we want to see. They include, there is temperature, we have light intensity, and carbon dioxide concentration. So those are the factors on which photosynthesis depends. Those factors have to balance. There has to be temperature, there has to be light intensity, and there has to be carbon dioxide concentration. So those are the ones now that which are going to act as our limiting factors. So photosynthesis as a reaction depends on those conditions. And photosynthesis, the rate of photosynthesis is going to be limited by those factors. Any of those factors which can be at its minimum value will be the factor that affects the rate of reaction or the rate of photosynthesis. Limiting factors set the speed. The limiting factor is the one that sets the speed. For example, we are seeing we are having temperature, we are having light intensity, and carbon dioxide concentration. So if we have the temperature is high enough and light intensity is enough, but we have carbon dioxide concentration is low, therefore carbon dioxide concentration which is low is the one which is going to be the limiting factor, and it is the one that is going to set the speed of the reaction. Temperature, light intensity, and carbon dioxide concentration are the possible limiting factors on photosynthetic rate. So let us look at the limiting factor of temperature. How does temperature affect the rate of reaction? So photosynthesis is controlled by enzymes, and that enzyme is rubisco. which is needed in carbon fixation. So we are seeing that when increased temperature will increase the kinetic energy of the enzyme, and this one increases the collision. And of course, when you increase the collision, then the rate of reaction is going to increase. So we can see that as the temperature increases, so does the rate of photosynthesis. Also, the rate of photosynthesis increases as we increase the temperature. So, at optimum temperature, the rate of photosynthesis now has reached its peak. So we have seen that as the temperature increases, also the rate of photosynthesis increases. Time reaches when we reach the peak. When we reach the peak, then the rate of photosynthesis becomes constant. At that peak, that temperature at that peak is what we refer to as the optimum temperature. At the optimum temperature, the rate of photosynthesis will reach a peak. So at this temperature here, we are seeing that now the rate of photosynthesis has reached the peak. However much you increase the temperature, the rate of photosynthesis has reached the peak. It cannot increase any further. So, higher temperatures will cause a decrease in photosynthesis as the enzyme is denatured. So, when we get our temperature and we increase the temperature beyond the optimum temperature, what is going to happen? The rate of photosynthesis now is going to decrease. Why is it decreasing? Because when you increase the temperature beyond the optimum temperature, now the enzymes are going to be denatured. Our enzymes are going to be denatured. And as you denature the enzyme, the rate of photosynthesis will go on decreasing. So now let us look at the limiting factor, which is the light intensity. We want to look at the light intensity as a limiting factor. Light is absorbed by chlorophyll and used to make ATP. And this one takes place in the light-dependent reaction. Light is absorbed by chlorophyll and it is used to make ATP, which is the energy. But as the light intensity increases, more chlorophyll becomes photoactivated. So this is going to increase the rate. So we can see from our diagram here that as the light intensity increases, what happens to the rate of reaction? The rate of reaction will also go on increasing as the light intensity goes on increasing. But after a certain point, the photosynthetic rate will plateau. That is, activity levels off. So as we have been seeing, increase in light intensity also leads to the increase in the rate of reaction. But after a certain point, then the photosynthetic rate now is going to plateau. Plateau meaning that the activity is going to level off. However much you increase the light intensity, the rate of reaction will remain the same, will not increase. That's what we are calling plateau. And this is because the enzyme responsible for carbon fixation, rubisco, are occupied. When we reach this particular light intensity here, at that particular point, all the enzyme rubisco, which is responsible for the fixation of carbon dioxide, carbon, is occupied. So however much you increase the light intensity, there will not be any further increase in the rate of reaction. Why? Because all the Rubisco enzyme is utilized or is occupied. Photosynthetic experiments. The rate of photosynthetic experiments or photosynthesis can be measured via the change in the reactants or the change in the products. That's when we can measure the rate of photosynthesis. We can use carbon dioxide uptake, and this one can be measured by the change in the pH. That is carbon dioxide that is dissolved in water to form carbonic acid. So we can measure the rate of photosynthesis by measuring how much carbon dioxide is picked up or taken. And this one can be measured by looking at the change in the pH. Because when carbon dioxide dissolves in water, it will form an carbonic acid, which means the pH will be low. And if carbon dioxide is used, that means that now the pH is going to rise. Then we can also use the oxygen production. How can we measure photosynthesis? By use of oxygen production. This one can be measured by the gas volume changes. For example, if we are collecting the gas, collecting it in a syringe, we can see the syringe size, volume increasing. Or we can use the rate of bubbling. That is counting the number of bubbles that are being given off. Those ones can be used to measure the rate of photosynthesis by using oxygen production. You can measure the gas volume change in the syringe which is produced, or you can measure the rate of bubbling. The rate of bubbling is telling us how much oxygen is being produced and bubbling out. Or still we can use the biomass change. In the biomass change, we are determining the changes in the dry weight of the plant. You get the plant and then you measure its change in the dry weight. That one can also give you the rate of photosynthesis. Thank you, my friend, for participating in photosynthesis. And today we have looked at photosynthesis. We have also looked at light spectrum. We have looked at chlorophyll. We have talked about chromatography. We have talked about the action and absorption spectrum. We have looked at the photosynthetic pigments or photosynthetic stages. And then we have looked at the limiting factor. Thank you very much for participating. And please do not forget to subscribe.